All right, whether or not the battery will last till the end of the lesson remains to be seen, but it's also the question whether your battery will last till the end of the session. Thank you for not all running off and coming back for the second part of this lesson, so I must be doing something right. I'm hoping to get all the way to the end of nerve impulses. We're almost there, all right? I can understand this is a slog. There's an awful lot for you guys to take on, all right? So, where were we? We know that we've got 100 billion neurons in our brain. We know that they're little fat bubbles, all right? They are fat bubbles that are filled with salt water. There's salt water all around them. They've got tube-like structures growing out of them. They're called axons and dendrites, okay? Now, neurons are filled with salt water and they are surrounded by salt water, but the type of salt water that's inside them is not the same type of salt water as that which is outside them, all right? And the reason for this is a protein, okay? And there's actually a whole series of proteins that will basically make it their job to pump certain types of salt ions out of the cell and pump others in, all right? So for example, all your neurons have a so-called sodium-potassium pump, which is a protein that will basically fold in a particular way to allow sodium ions, okay, sort of cooking salt ions, to stick to it on the inside, all right? And when they're stuck in there, it'll flip over, all right, to let the sodium ions escape, and then it'll wait for potassium ions to bind, and then it'll flip back in and let the potassium ions in. And like this it will basically, and this is a process that is actually driven by your cell. It actually expends energy on pushing out sodium and sucking in the potassium. All right. And the consequence of this is that the fluid outside your cells is very high in sodium. All right. So this is like, that's why your tears are salty. Because you've got these pumps pumping sodium out and pumping potassium in. All right. There are similarly, other pumps, the pumps that will allow sodium back in order to push calcium out, all right? So calcium is very, very, very low inside your neurons, okay? Your neurons are high in potassium, low in sodium, low in calcium, and they're also low in chloride, okay? Chloride also gets pushed out of the cells, okay? Now this is so we have uneven distributions of different sort of ions here, so that sodium is low inside, high outside, chloride is low inside, high outside, calcium is very low inside, higher outside, potassium very high inside and low outside. These particular values, if you're trained to be a medical doctor, you have to know them, all right? You're not training to be a medical doctor, or at least not yet, all right? So you don't have to learn them, but you should, if you possibly can, remember that the fluid that bathes the cells of your body, including your brain, is largely saltwater solution, sodium and chloride in water, all right? Whereas there's much less sodium and much less chloride inside the cells. And inside the cells you get lots of potassium and lots of organic anions. These organic anions come from the proteins, okay? So we've got sodium chloride outside, potassium organic anions inside. Now that's the status quo, all right? And we've got very little calcium. That's the status quo, that's the way the cell normally is. But of course, the thing is, all of these sodium ions and the chloride ions and the potassium ions, they're constantly being bounced around by this water that kicks into them with all this sort of fervor that I talked about earlier, So if they get a chance, if the sodium ions get a chance to to go in, or the potassium gets a chance to escape, they will use that chance. And the force that drives this is simply the diffusion that I explained earlier, all right? So, so of course, normally they can't do this unless there are pores, okay? But unless there are sort of channels through which they can go. But in fact, there are channels, and the reason that there are channels creates a voltage, an electrical driving force across the membrane of the cell. If you were to stick a little electrode, and people can actually do this as an experiment, stick a little electrode inside a nerve cell, and you measure the voltage relative to the outside of the cell, you realize that there is actually a voltage, which really just means it's an uneven charge distribution. Okay, so that you get more positive charge on the outside of the cell and more negative charge on the inside of the cell. Now, of course, these positive charges will attract the negative charges, and it causes like a sort of, you can like a tension, okay, that will actually sort of that you can measure in volts, all right, and it's about 65, 70 millivolts or so would be, for the nerve cell at rest, would be the voltage across its membrane. Now 60 millivolts doesn't sound like much, and it isn't all that much, because if you buy yourself a little battery, it'll be 1.5 volts, okay? That's substantially more than the 60 millivolts. But you have to bear in mind that these 70 millivolts or so operate over a membrane that is only about 10 nanometers thick. The thing about electrostatic attraction between charges is that it obeys what physicists call the inverse square law. Now that sounds very complicated, but ultimately it just means that the force is very strong if things are close together and not so strong if they're further apart. Okay, and the force changes with the square of the distance. If you've ever played with magnets, if they're far apart, you don't feel much, but if you try and get them really close together, then the attractive force or the repellent force suddenly becomes very strong. All right, this is sort of also due to the inverse square law of the magnetic forces. All right, so similarly the electric forces, the positive and negative charges that are unevenly distributed across the cell can attract each other, can have a very strong attraction here. It corresponds to 7 million volts per meter electric field strength. Now, in air, an electric field strength of 3 million volts per meter is what it takes to cause lightning. All right. So on the very small scales of the membranes of your brain cells, this piffling little 60 or 70 millivolts is a huge electric force, which if you happen to be just a tiny little protein that sits there, you will feel it. It will bend you out of shape. All right. So a lot of just by distributing charges differently across the membrane, we can do things on the very small scale that matters for neurons. So this was just to remind you about the electrostatic forces that really create this attraction and repulsion. So really, what happens in a neuron at rest is that it'll have a lot of potassium inside it, very little potassium outside it. The potassium will want to diffuse out if it can. Okay. Now all your neurons in your brain will have little channels in them made out of protein that will allow potassium to leak out. Okay, and this potassium that will leak out will therefore means we get suddenly, but of course potassium is positively charged, so we suddenly get a surplus of positive charge on the outside and negative charges from the organic ions left behind. Now there will be an electrostatic attraction. They will try and suck the potassium back in. So we've got two forces opposing each other: diffusion pushing it out, electrostatic attraction pushing it back in. And there'll come a point, the so-called equilibrium potential, where where on average you get as much potassium being expelled by diffusion as you get sucked back in by electrostatic attraction. Okay. And that's what creates this electric tension across the membrane of the neuron at rest. Okay. All neurons at rest will have a charged, polarized membrane. Now the thing is that that's how a neuron basically says, okay, there's nothing happening. My membrane, my outer membrane, has this voltage applied across it. Nothing's happening. That's my status quo. But of course, you could imagine what would happen if you. All right. The first thing that we reach that point, okay, where you get that electrostatic equilibrium after only relatively few potassium ions will have leaked out. All right. So if we start off with a distribution where there's 10 times more potassium in than out, and then I open pores that allow the potassium to escape, I get that voltage difference. No more escapes. It'll still be almost 10 to 1. All right. Very, very little potassium escaping can generate a relatively large voltage because the electrostatic forces are actually very strong. Okay, that's what this little slide is all about. Now, physicists have a complicated formula that involves gas constants and temperatures and Faraday constants and stuff, where they can calculate exactly where, for a particular ion distribution, concentration and so on, that what voltage you need to reach before you get to that equilibrium point. All right. We don't really need to worry too much about this. But the thing is that ultimately, what I want you to, if I've got a lot of potassium inside and I get potassium leaking out to create a voltage, well, I've also got a lot of sodium outside, which is also positively charged, which will want to go in. All right. So why does the sodium not go in and cause a reversal of this? All right. Why is potassium the one that matters? Well, it turns out that a neuron normally has these, this particular protein here, the so-called potassium leakage channel that's in all neurons, is a protein that will only allow potassium through and nothing else. Okay, and it can do that basically because with these hydration shells around them, they are different in size, and it can kind of, it allows potassium to to shed its hydration shell, go through, acquire another hydration shell here. Okay, and sodium can't do that; it can't go through the same channel. But I can have another channel which I can open and close at will, which might allow sodium back in. All right. So if I allow sodium back in, then of course the sodium will suddenly say, oh look, I've got electric attraction here through my negatively charged ions, and I've got a concentration gradient pushing this way. And then suddenly this voltage that we had across the membrane will collapse, and the voltage will disappear. This is what people call the neuron becoming depolarized. So when a neuron becomes depolarized, this is actually somewhat oddly, is the excited state for the neuron. The smaller the voltage across its membrane, the more excited it is. All right. And the thing is that what happens is that a neuron will normally be highly permeable only to potassium, but it may, under certain circumstances, permeable meaning that potassium can go in and out of the membrane, all right? But it can, under certain circumstances, become permeable to sodium, means that it allows sodium to flow through. And if that happens, the voltage across the membrane collapses, okay, and the neuron becomes excited. So what I want you to, forget about that text. You can read that later, all right? It's on the web. You're a neuron, all right? You're a little fat bubble. You've got potassium inside. You've got sodium outside. You're at rest. You've got lots of little channels that let the potassium go out, and therefore your membrane is under tension. All right, ready for action. Okay. Now something happens. All right. Perhaps you're a stretch receptor in the skin. All right. Somebody pulls on you. All right. The pull pulls open proteins that sit in your membrane that are little proteins that will, because they're now pulled, will open a little channel through which sodium can rush in. The more you pull, the more sodium rushes in, and the more you lose your polarization, the more depolarized you become. Your voltage goes from being minus 70 millivolts to minus 50. You pull some more, it goes down to minus 30. You pull some more, it goes down to minus 20. You pull some more, it may go all the way. Actually, if you let the sodium completely overtake the potassium, become the dominant ion that flows much more than anything else, the polarity will actually change and will become positive on the inside relative to the outside. Okay. So no, negative on the inside relative to the outside. Sorry. So so you've got you can basically start to encode the world around you, and this is a really important concept. Okay, by how strong the charge on your membrane is. If you happen to be a stretch receptor in a neuron's skin, then the more depolarized you are, the stronger the stretch. All right. If you are a light receptor in your eye, actually, light receptors, we'll talk about them, 2 lessons from now, you will signal how dark or how light it is by how depolarized your membrane is. All right. If you're a receptor in the ear, you'll become more or less polarized depending on how much sound pressure there is, and so on. So you encode the world around you through membrane voltages, and this is something that you might call an analog code. Analog because there's an analogy between something that's out there in the world that might interest us and the voltage across your membrane.